Analytical method validation and analytical method validation parameter is the topic for the current video, and let's start with this video. Welcome dear viewers to the video on analytical method validation. Analytical method validation, it is also abbreviated as AMV. AMV activities are very important in the pharmaceutical field, and AMV is required for ensuring the quality, safety and efficacy in the pharmaceutical products through the quality testing. It is a cornerstone of regulatory compliance and the product excellence. So, as we know that the pharmaceutical formulations, the pharmaceutical products are required to have the required quality, safety, and efficacy. And these pharmaceutical products, including the raw material, packaging materials, and also the finished formulations, should meet the quality parameters. So some testing technologies are there, some testing activities are there, some analytical testing procedures are there. So these are required to meet certain criteria so that the testing methodology results can be reliable, and that's why AMV activities are performed. AMV activities are essential for confirming the accuracy, precision, and reliability of the analytical procedures. If you are performing the testing for potency or assay by some testing procedure, and if you come to know that that testing procedure is not proper, then it means the results whichever are obtained are also not proper. So AMV ensures that the pharmaceutical products meet regulatory requirements, safety standards, and maintain the high quality. That means the testing procedure is reliable. Analytical method validation is a key for producing trustworthy results and maintaining the patient safety. So just think that you tested the formulation for assay. The assay is confirming to the standards like 90 to 110%. It is coming as per the label claim between this window of 90 to 110 or 95% to 105%. But if you check the same product once again, and if it is failing, then what is the problem? That means the product is same, but the analytical method is not proper. That's why validation is required. Validation is important for quality assurance, regulatory compliance, patient safety, and excellence commitment. Based on the analytical testing, the product is released into the market for use by the patients for the disease treatment. And think that if the analytical method is not proper, what will happen? Sometimes the default product or faulty product or the failure product is getting released into the market. So for quality assurance, the validations are important. Validation ensures consistent and reproducible results. Also, regulatory compliance is required to meet industry standards and legal requirements. Patient safety confirms safety, efficacy, and high quality products. Excellence commitment beyond regulatory requirement ensures the best possible products for the consumers. So the product should meet the basic criteria and as well as the product excellence. Come to the key parameters in analytical method validation: accuracy, precision, specificity, linearity, range, and many others like robustness, ruggedness. We will see these in the upcoming slides. Accuracy is the closeness to the true value or standard, how much accurate the results are. Then precision is the consistency of results when the method is repeated. Then types of precision: repeatability, precision under the same operating conditions over a short time; intermediate precision, precision across different days, analyst or equipment; reproducibility, precision between the different laboratories. And why this is required? Because the analytical methods are developed in the research lab and then these are transferred to the quality control labs. So there should be accuracy, precision, repeatability, intermediate precision, and reproducibility. Specificity is the another parameter. Specificity is ability to assess the analyte in the presence of other components like impurities. So the method should be accurate, it should be precise, and it should be specific. Linearity. It is direct proportionality between analyte concentration and the results. For example, you are working on the formulation containing 100 mg of the API. So you can check the linearity by having different concentrations like 50%, 100%, and 150%. So linearity should be there. Range. Range is the interval between the upper and lower concentration where the method is accurate, precise, and is linear. So for range, you should show that the upper and lower concentration measured by the method are accurate, precise, and linear. Some parameters are there which are tested in the validation, and validation is performed on these parameters, you can say. Robustness, ruggedness, LOD, LOQ, recovery, system suitability, selectivity, stability, intraday, interday. Many parameters are there. We will see those parameters one by one. So robustness. So method should be robust. That means it should be able to withstand small changes in the parameters like pH and temperature. It is not like that if pH is set 5, if it goes to 5.1 or 5.2 and then the method is showing some drastic results. It should not be like that. Ability should be there to withstand small changes in the parameters like pH, temperature, and others. Because if the method is not robust, you will get different results different times. Ruggedness is the another parameter. Ruggedness is the degree of reproducibility under normal operating conditions. If you check the product or you test the product for some test, once again you test it in the similar conditions, then the results should be within certain range. It should not vary much. So ruggedness is the degree of reproducibility under normal operating conditions. Then limit of detection, that is abbreviated as LOD. So LOD is the lowest concentration detectable but not quantifiable. Understand that, remember that LOD is the lowest concentration detectable but not quantifiable. You can detect that but you cannot quantify. Then come to the limit of quantitation, LOQ. Lowest concentration that can be determined with acceptable accuracy and precision. LOQ is the terminology which is very important to understand, and as per this definition, LOQ will always be more than the LOD. LOQ is lowest concentration that can be determined with acceptable accuracy and precision, because LOQ concentration will always be more than LOD. Then is the recovery. Percentage of analyte recovered during extraction indicate the method efficiency. Suppose that if you are adding 10 mg to the solution and you are not getting the 10 mg, that means recovery is not there. Recovery is percentage of analyte recovered during extraction indicates method efficiency. Then system suitability. System suitability means the system works correctly, and system suitability ensures the system work correctly before or during analysis. If you are working on some system like HPLC, then system suitability test should be there. Selectivity, ability to differentiate the analyte from the substances which are other than the sample. For example, if you are making a tablet of aspirin, or paracetamol or diclofenac or telmisartan. You are using some excipients to make the tablet, and the analytical procedure should be such that it detects the active material so that it differentiates the analyte from the placebo material. Stability. Stability of the analyte in the sample matrix under specific conditions. Example, storage, processing. So the analyte should be stable. So as we have seen the parameters for analytical method validation, AMV is a very critical important phase in the pharmaceutical product life cycle and is a cornerstone of pharmaceutical quality assurance. It ensures products meet the regulatory standards with accuracy, precision, and safety at their core. AMV provides the confidence for the analytical method that it will give the precise and correct results. It provides a commitment to excellence, reliability, and patient safety. So this is the information regarding analytical method validation and the validation parameters. Let's meet soon with such kind of informative video, and I request you to share these videos to your friends and colleagues. Thank you.